So it's a pleasure to introduce Jason Fang from my lab. He is a senior graduate student, and he grew up in Irvine and went to UCLA, where he worked for Yi Tang, and also played in the marching band there, the drums. And so, as you guys might know, UCLA is a pretty big band school, so it's I think it's a very interesting accomplishment. And he has been just a wonderful person to work with, just accomplished a lot, and just everything that he will talk about here today is really his own ideas and his own work. So I'm just really happy to be here, and I'll just give him his time to talk. So Jason, if you want to get started.
Thank you, Michelle, for that wonderful introduction. And thank you to the organizers of this meeting for giving me the opportunity to talk about some of my work on the aldolase-catalyzed synthesis of chiral organofluorines. So as we all know, enzymes have become really important tools for synthesis of drugs, since they're extremely efficient and selective. And some of the aspects of biocatalysis that have been traditionally considered as cons, such as a limited reaction diversity and substrate scope, have basically become non-issues in the modern age due to advances in things like directed evolution and bioinformatics. One of my favorite examples of successful industrial biocatalysis is the case of sitagliptin, or Januvia, a blockbuster anti-diabetic drug produced by Codexis and Merck, using this a highly engineered transaminase enzyme to synthesize a chiral amine in exquisite stereoselectivity. And the development of this biocatalytic process streamlined the entire synthesis by eliminating the need to perform a series of costly purification steps. So, so now we know that enzymes can are a great use for synthesizing drugs, but when we actually look at okay, I think my slides are automatically advancing. Okay, I'm just gonna use the not full screen mode because I don't know how to stop my slides from automatically advancing. Okay, but when we look at the structures of drugs that are commonly approved drugs, we find that a large number have approximately a quarter of drugs contain at least one fluorine atom. And globally, there's over 300 fluorinated drugs. In contrast, in the field of natural products, there's very few fluorinated natural products, less than 20, and only one known enzyme that can actually form a carbon-fluorine bond. That is the fluorinase, which is actually responsible for making those natural products. So comparing these two sides, we see that although fluorine is very abundant in pharmaceuticals, it's super rare in nature, and thus there's this large gap between fluorine chemistry and biocatalysis, which is what we would like to bridge in our group. Some of the properties that make fluorine very attractive as a drug motif derive from the fact that it's a suitable steric replacement for protons and hydroxy groups and the like. But it's actually, due to its very high bond dissociation energy, the CF bond is also considered the strongest organic bond. Additionally, the high electronegativity of fluorine confers the ability to tune the protonation state, the conformation of drugs, as well as interacting with proteins and blocking adventitious metabolism of drugs by enzymes such as P450s. Selective fluorination, incorporation of fluorine into drugs, is often difficult and expensive, and this drives our interest in looking for a biological approach. So in the first part of the talk, I'll start talking about our discovery and characterization of a mode of reaction that allows us to use fluoropyruvate as a substrate in enzymatic aldol additions. In the second part, I'll talk about a more recent extension of this work to synthesize these fully substituted quaternary carbon-fluorine stereocenters. So with the first part, I want to begin with some brief background on aldolases. Aldolases are enzymes that in vivo perform the retroaldol cleavage of metabolites in the catabolism of sugars and some xenobiotics. Here I'm showing the example of a pyruvate aldolase, which is cleaved into the nucleophilic substrate, or the donor, which is pyruvate, and various acceptors, which are usually highly polar or negatively charged aldehydes, such as glyceraldehyde 3-phosphate. It's been known since around the 1980s from the pioneering work of people like George Whitesides and Chiwei Wang, that aldolases can be used in the reverse direction by supplying the donor and the acceptor in order to form the carbon-carbon bond with formation of up to 2 new chiral centers. And as shown by these variously colored spheres, if we're able to engineer substrate promiscuity into both the donor and the acceptor, we can have a very flexible system for carbon-carbon bond formation. And specifically, we're interested in putting fluorine atoms at the alpha position of the donor in order to make a new fluorine chiral center. So to start off, the main problem with these approaches is that the donor specificity of aldolases is typically strict. On the other hand, people usually find that the acceptor specificity is quite promiscuous, so that's not as much of a problem as mostly a solved problem with directed evolution and things like that. But the donor is different, and donor substrate scope expansion has been mostly the modern focus. So our focus is going to be, can we take a pyruvate aldolase and use fluoropyruvate in its place? A further motivation for us for using the aldolase approach is in these anti-cancer and antiviral nucleoside anti-metabolite drugs that contain a fluorinated sugar in them. And in these types of drugs, the regiochemistry and the stereochemistry of the fluorine atom are critical for the bioactivity of the molecule. Since these sugars all contain a vicinal fluorohydroxy moiety, we thought that they would be suitable for an aldol-based approach. So looking in the literature for some precedent, we looked in the literature for any precedent where fluoropyruvate was used as a substrate, and it turned out that there were a few limited cases where it did work. And in these 2 precedent papers that I'm showing on the left, pyruvate aldolases were coaxed into utilizing fluoropyruvate, albeit with very limited aldehyde scope, and also these enzymes, because of the mechanisms of these aldolases, which utilize a catalytic lysine residue to form a Schiff base with the donor substrate. These enzymes are susceptible to inactivation of the lysine at high concentrations of aldehydes. And of course, in any industrial process, we would like to be able to use a very high concentration of aldehyde. So we started from there and started to do our own screening of pyruvate aldolases. And eventually we stumbled upon this family of pyruvate aldolase called HPCH, which, unlike these previously mentioned type 1 aldolases, the HPCH family is a type 2 metal-dependent aldolase, so it doesn't suffer from these previously mentioned drawbacks. And we found that this family of pyruvate aldolase can effectively utilize fluoropyruvate under a variety of conditions, and in work that was published in 2019, which I'll go into a little bit of more detail now. So our assay is very simple. We mix aldehydes, a pan of various aldehydes with fluoropyruvate in a buffer and let it sit overnight, and then we take fluorine NMR. So not only can we tell simply by integrating the fluorine NMR the extent of conversion, but we can also see the formation of syn and anti-diastereomers. I forgot to mention, we add hydrogen peroxide to the products to simplify the spectrum, since the removal of the C1 carboxylate results in a simplification of the NMR spectrum. So we did this analysis for several different homologs of enzymes within the HPCH family. And for here in this bar graph, I'm showing the data for 3 enzymes, with each of these 12 letters corresponding to a different aldehyde substrate. And within each group of 3 bars is plotted the diastereoselectivity, the amount of anti and syn products for each of the 3 enzyme homologs that I'm showing. And I just want to make 2 points about this. One is that although you can see that there's quite a diverse range of aldehydes within the set of 12 aldehydes, the first 8, which are polar, still work much better than the last 4, which are more non-polar. And the other point I want to show from this bar graph is that the diastereoselectivity is sometimes high, or is sometimes highly selective and sometimes non-selective depending strongly on which aldehyde and exactly which enzyme homolog you're using. For example, in the substrates 1B and 1D, the reaction is highly anti-selective no matter what. But for some cases, like aldehyde 1G, switching from one enzyme homolog to the other for some reason actually can give you almost entirely the anti-product or almost entirely the syn product. Okay, so I might get into the reason in like 2 slides, I think. But a little side note on the kinetics of the reaction. As I noted before, we performed this standard assay by letting the reaction sit overnight. And when we actually take a time course to investigate kinetics, we see that the reaction with fluoropyruvate marked by this red line is actually proceeding slowly overnight. If we were to, however, switch the substrate back to just the native pyruvate substrate, we find that the native pyruvate donor, we find that the reaction is actually pretty much complete within a couple minutes if the same amount of enzyme is used. So it shows that the presence of a fluorine atom confers a significantly slowing down of the reaction. Using a discontinuous assay with a mass spec detection-based approach, we were able to get some Michaelis-Menten kinetic parameters for pyruvate and fluoropyruvate as aldolase donors. And we find that the slowing down with fluoropyruvate is primarily attributable to a 500-fold defect in the kcat, the turnover number, with not too much of a difference in the km. So now going back to the issue of, let me go back 1 slide just to mention. Now I'm going to talk a bit about the stereoselectivity, which, as I mentioned, the diastereoselectivity can result in anti and syn products. But when we investigate the absolute configuration, that is whether these are R or S, it turns out that the fluorine atom is always, as shown here, the fluorine is always S configured, while the anti and syn product distribution results from cases where the hydroxyl group is not defined and can be either be one configuration or the other. In order to probe why this was the case, I collaborated with my friend Diptar Kahait from the Martin Head Gordon lab here at UC Berkeley to perform some DFT calculations on this reactive intermediate species that is this fluoroenolate intermediate chelated to a metal in the active site of the enzyme. And what Dip found was that this reactive intermediate can theoretically take one of 2 forms, either the cis fluoroenolate or the trans fluoroenolate, but he found that the cis fluoroenolate, or the one with a Z configuration, is vastly more favorable in energy compared to the trans or E configured enolate, which, as shown by these models, potentially suffers from some kind of severe electronic repulsion. So the reactive intermediate we propose in the fluoroaldol reaction consists entirely of this cis configured fluoroenolate, which then can go on to attack an aldehyde. And because the aldehyde is not recognized by, is not specifically bound by many residues in the enzyme active site, it does not really have a preference on which face of the aldehyde gets exposed to the enolate nucleophile. And that results in having the potential of anti and syn products where the hydroxyl group stereochemistry differs, but the fluorine stereochemistry is always fixed. So in summary, the fluorine stereochemistry we found is fixed by the architecture of the active site and the fact that the enolate is always a certain geometric configuration, while the hydroxyl stereochemistry often lacks control. But in some other data that I'm not going to show here for sake of time, we actually showed that the hydroxyl stereochemistry can be improved or reversed by active site engineering. We decided to take this newfound knowledge and show the robustness of this enzymatic system by scaling up some of these transformations. For the first 6 compounds I'm showing here, we isolated these highly polar fluorinated sugar acid type compounds by preparative HPLC on a scale of like tens of milligrams. And then for the last 4 products I'm showing here, we performed these on a near gram scale. After applying proper protecting groups, they could be purified simply by organic extraction. We were also inspired by the reactions that pyruvate itself can undergo in nature. That is, in nature, pyruvate can be transaminated to give alanine, and it can be reduced by a lactate dehydrogenase to give lactate. To try out some of these reactions, we obtained transaminases and lactate dehydrogenases and found that in many cases, they were able to transaminate and dehydrogenate our intermediate fluoropyruvate aldol adducts. In that case, giving us various 3-fluoro-2-hydroxy acids and some novel 3-fluoro-2 amino acids, such as these compounds in the box. Additionally, taking some of the compounds from the left side and then performing chemical reductions on the C1 acids, we were able to obtain various fluorosugars, which harken back to those fluorosugars that are present in important antiviral drugs. So hopefully I've been able to show you with this first part that we have a pretty diverse platform for making more complicated molecules from simpler fluorinated compounds. And yeah, so this is just our kind of schematic for summarizing the possibilities from the fluoroaldol adduct. And with that, I'll move on to our more recent work on extending this work to synthesize all quaternary carbon fluorine stereocenters. So quaternary carbon fluorine stereocenters are present in some very important drugs, especially as I've shown several times in this presentation, the nucleoside mimic sofosbuvir, which is has been the subject of a number of chemoenzymatic syntheses. However, these 3 drugs that I'm showing here is not actually just a subset. It's not actually a subset of quat CF stereocenter drugs. It's actually these 3 drugs are the only approved drugs that contain quat CF stereocenters, and the reason that there's so few of them, that they're so underrepresented in drug candidates, is mainly due to a lack of synthetic methods. We feel that we can use our aldol-based approach by simply graduating from using fluoropyruvate as a substrate to using fluoropyruvate with this additional alkyl additional substituent shown by the green sphere. As a side note, this novel substrate, this substituted version of a fluoropyruvate, we would have prepared it in a racemic form, and within this racemic form, only one of the enantiomers would be a competent substrate for the enzyme. This is actually a kinetic resolution, but that doesn't have too much bearing on the nature of the product, besides the fact that it leaves the unreactive enantiomer of the substrate here. So we wanted to test this with a number of new fluorinated donors that are basically longer, more bulky analogs of fluoropyruvate. These are not commercially available, so we had to briefly figure out how to synthesize them. I'll go into that quite briefly. So we can make most of these fluorinated keto acids by starting with a non-fluorinated keto acid or an ester, forming the silyl ether, and then treating that with select fluor, which can then be saponified to give the desired substrate in its sodium salt form. This synthesis had parts and bits of it mentioned around in the literature, but in order to optimize this on a gram scale, we spent some time tweaking the conditions, and coincidentally found out that everything works really great if you just do every step of the synthesis in acetonitrile, which was pretty funny. This last substrate, which contains a phenyl group substitution at the 3 position, was synthesized according to a literature procedure, with the key step being the opening of this epoxide with HF pyridine to give the 3 fluorinated compound. So now that we had these substrates to test, we wanted to see what mutations could be made to the active site of HPCH and family aldolases in order to accommodate this new steric bulk. So here I'm showing 2 different representations of the active site around the active site of HPCH. And a lot of this mutational work was done with Laura Turner, who is a great exchange student from University of Oxford who worked with us for a year. So as you can see, situated around this pyruvate molecule are these tryptophan, leucine, tryptophan 19, phenylalanine 170 residue, and leucine 212 residue, which are the primary steric determinants on whether the groups attached to the pyruvate can be accepted or rejected. There was this interesting report from 2018 that the tryptophan 19 reduction was absolutely essential for accommodating non-fluorinated bulky donors. However, when we tested it with our fluorinated bulky donors, we found that the W19 mutants, such as W19A or W19V, were totally useless. And going back to the drawing board, we realized that we actually had all the information to figure out why this was the case, as I'm showing in these little schematics here, that when the donor substrate only has a fluorine substitution or only has an alkyl substitution, both of those want to fight for the cis position and therefore are related to bumping into the W19 residue. However, when we have both a fluorine and a bulky alkyl substitution, the fluorine wins the fight for the cis position, and therefore we hypothesize that F170 and L212 may actually be the correct mutations to make. So we went back to a similar version of the assay I mentioned before, where in this case, we will incubate these bulky donors in the presence of formaldehyde and test it with all the variants of HPCH, and then perform the same assay. And just as a summary of what the results were, we found that these three substrates with just regular fluoropyruvate or having a methyl or ethyl group substitution were accepted by a wild type enzyme or by a mutant, whereas these last three that I'm showing here were hitherto have been, we have not been able to incorporate them, or actually this last one with the phenyl group, we found that it was unstable in our reaction conditions. So I won't talk about those three any longer, and the next data that I'll show is just about these first three substrates. So in this table, I'm showing all the active site mutants that we tested with these three substrates and the extent of conversion. And what we notice is that all of these mutants were actually less active when tested against the small donors, as you can see for just fluoropyruvate and the one with the methyl substrate, wild type enzyme was actually still better than any of these mutations. So these mutations confer actually a decrease in the intrinsic activity of the enzyme. However, when we moved to the bulkier ethyl group substitution, then we found that the F170 or the L212 mutations were in fact superior to the wild type. After doing this table, we kind of settled on one optimized condition for each of these donor substrates and scaled up to a single optimized condition in order to perform testing of their aldehyde scope and scaling up reactions. So with all this knowledge, we were able to expand our substrate scope from before to include a wide variety of aldehydes, including such as extremely nonpolar, this cyclohexane example, for example, and then a wide variety of heteroaromatic and aromatic examples. And so the way to basically read all this stuff is that if there's if the number below the compound has the numbers 4, 5, and 6, then it worked with all three of these donors. Whereas if it just says a 4, then it was actually just done with only fluoropyruvate. So as a last part of showing the utility of the system, we wanted to isolate several of these examples that have potential as drug fragments. So for example, we're able to isolate several analogs of this 3-hydroxy ester, which was used many times in a classic total synthesis of the compound of the anti-cancer compound discodermalid. Some of these aromatic compounds appear to be bear some resemblance to drug motifs carrying these stereotriad motifs with the aryl group. And additionally, we were able to make now methyl branched fluorinated sugars as well, which may be useful for incorporation into these fluorinated fluorinated nucleoside drugs. So in summary, I hope I've been able to show you that the HPCH pyruvate aldolase catalyzes efficient aldol addition, and we investigated its substrate scope, its stereochemistry, and its kinetics. These products can be isolated or subjected to downstream chemoenzymatic transformations. And more recently, we've been able to show that the donor scope is expandable beyond fluoropyruvate to a select few slightly bulkier fluorinated keto acids, thus providing access to quaternary carbon fluorine stereocenters. So with that, I'd like to thank my mentor, Michelle Chang, as well as all members of the lab and the alumni who have all helped me a great deal. I'd like to thank Dib Tarkahite and Martin Head Gordon for the DFT calculation part, and the NMR facility at the College of Chemistry here for all their help. And with that, I'd like to take any questions.
Okay, thanks very much, Jason, for a fantastic talk. So is it Andrew or Anya that's going to coordinate the questions?
I'll take this. So Anya tells me that we have a question from Hao Yang at the Protein Engineering Department here at Merck. Would you like to answer live? I've given you permission for your microphone. If not, I can ask the question for you, Hao. I'll take that as, oh, I will ask, okay. Hi, Jason, could you comment on the availability and any potential mutagenetic toxicity for fluoropyruvate? I think these are some concerns for its application in large scale synthesis.
Right, that's a great question. So I don't have a good idea of how exactly toxic it is because I haven't done any of these experiments with cells. I always use purified protein. Fluoropyruvate is not, I don't think it's super toxic. I take basically no precautions when working with it. But it is known to be an inhibitor of pyruvate dehydrogenase, which is a central enzyme in central metabolism. So it may have some effect on that. But I feel like if the yeah, so I guess my answer is it is kind of toxic, but not super. So I think it may be able to use it in vivo format somehow. Maybe you can circumvent pyruvate dehydrogenase. I don't know.
Great. Okay, we have one more question here from Peter. Did you manage to identify the region on the enzyme you can mutate so that the stereochemistry of the hydroxyl group is controlled?
Yes. So the region on the enzyme is actually those same three residues that I mentioned. So this enzyme is kind of interesting if you were to take a engineering approach in the fact that there's actually not, it's kind of obvious that when you look at the active site that there are no other very involved residues. It's kind of a very remarkably simple active site. So whether you want to control the donor scope, if you want to engineer the donor or engineer stereoselectivity or engineer the acceptor, I think the primary active site residues are all the same three, just those three, and then any other improvements would have to be made by non-rational engineering outside the active site.
Great. So we have two more ones that just came in. Have you observed any defluorinated side products during the reaction?
So defluorination is very little. Routinely, I see 2%, around 1 to 2% defluorination. So it's very little.
Great. And one last one here. Do you think this is the best route for bringing fluorochemistry in general via biology, like to new nature fluoroxenobiotics?
I think that this is a good starting point, if not for the fact that fluoropyruvate is an easily commercially synthesized and commercially purchased compound. But I think it would be great if, despite that, the pyruvate aldolases themselves are not super well known, and I don't think they've really been used in any landmark preparations of landmark chemoenzymatic preparations of anything. So I think it would be great if it could also be just expanded to completely, if the fluorine biochemistry could just be completely expanded to other types of reactions as well, I think that would be great.